Welcome to New Brunswick Wildlife Wonders, a program that will highlight some of the projects funded by the New Brunswick Wildlife Trust Fund. I'm your host, Tom Benjamin. Today we'll be heading to the Grand Lake area where we'll be looking at the Bioblitz initiative, a project that occurred earlier this summer and looked at a variety of species in that area. Let's head there now.
Bioblitz is an effort to inventory all the organisms, all the living species usually in a defined geographic area. It's usually a protected area of some kind, a municipal park, a national park. In this case, we're working in New Brunswick's 10 largest protected natural areas. There isn't actually a hectare on the planet that scientists can point to and say that we know every species of fungi, green plant, and animal that occurs there. So we've got about 11,000 hectares here at Grand Lake Meadows, so the task is really an impossible one, but we are trying to cover as wide a variety of taxa, of plants, animals, fungi that we can, and we're trying to provide a baseline for future monitoring of both these sites and the larger New Brunswick landscape. So the question is quite a simple one. It's what's out there, but the answer obviously is a very difficult one to provide. You're going with Reggie, 11 o'clock,
Yep.
right? Okay, so we just still wait for Reggie to turn up. We don't have all the experts that we need here in the Maritimes, so we're very fortunate that we're able to bring together folks from right across North America. This year, in fact, we have our first international participant. We have a lady here who is a specialist on mollusks who's come from France. Generally, we've had people from really right across the U.S. and Canada. We've had a few regulars who come from, we've somebody from Texas, Kansas. We've got someone here from Maine this year studying slime molds. Have people from Ontario, Nova Scotia, and throughout the region. So we've got a wide range of people here, and many of whom have come quite some distance. This is a common field cricket. There's actually been very little work done on the grasshoppers of the region. The entire Bioblitz program is laid out as a 20-year project. We spend 2 seasons in each of the 10 largest protected natural areas. So we started in 2009, so we still have a good ways to go, but this is our third protected natural area that we're finishing up. Typically what we do is spend 2 weeks during that first year in late June, early July, working in the site. And then in the second year, we arrive in the latter part of August. There are, of course, different plants and flower, different insects flying. So we're trying to cover as wide a cross-section of the biological diversity in these sites as we can, and we can do that by trying to sample throughout the season as much as possible.
So you got that one? One of our objectives for the Bioblitz here at Grand Lake PNA is to document the diversity and abundance of small mammals in the small mammal community assemblage. So right now we're sampling this part of the island using snap traps for the New Brunswick Museum. And essentially what we're trying to do is catch all the small mammals that reside on the forest floor here, in all the nooks and crannies that you see, in all the mouse holes, and all the runways. So that's what we're doing here.
One thing I'm interested in looking at is how the mammal fauna has changed from last year. So last year's Bioblitz was done in June, now we're doing it in August. Generally we see an upward trend, so we get more mammals in August and June. So we want to see if we've gotten more again, and if we have any different species than what we caught last year.
The reason why we employ snap traps and the reason why we set them out all day is because a lot of small mammals are nocturnal. So that means they're most active at night. So we set the traps during the day, we leave them out overnight, and then we recheck them the next day in the morning to see what we've actually caught. It's a little physically exerting sometimes. It's long days, a lot of hiking, but I thoroughly enjoy it. Here in New Brunswick, we've recently actually detected a close relative of one species of mouse. We actually have one native mouse here called the deer mouse that belongs to the genus Peromyscus. We recently detected a close relative of the deer mouse on Grand Manan Island, and one objective here is to see if we can actually find that close relative, the white-footed mouse, also in the same genus, here in this part of New Brunswick. And the second objective for studying deer mice is that they are actually an important vector for transmitting Lyme disease. So a lot of our work applies to disease ecology and human health and wildlife management.
If we don't know what we're protecting, it makes it less valuable to the public. So if we know that there's a species here, for instance, that has never been described by science, as Reggie has found with beetles, that makes it more important in the public eye and more likely that this area will continue to be protected and not developed. So if you don't know what you have, it's harder to protect it and manage for it. So finding out what these species need to survive long term would be very helpful for conserving the natural world in New Brunswick.
The Bioblitz program grew out of some discussions that were held by the Scientific Advisory Committee on Protected Natural Areas. I'm a member of that committee and offered to organize our first Bioblitz, and the museum has since then really taken over the role of coordinating this program. So the funding for this it all comes from outside the operating budget for the museum. So I look after raising that money each year and bringing the people together who are going to participate. So the museum really is organizing this and has continued to develop the program over the last couple of years.
You are not thinking of driving, are you, my friend? Come now, Stuart. Reconsider. You're in no condition. Ring for a taxi. I'm telling you. Don't be a fool. I'm begging you. Trust me. You are high. Way too high.
Tune in to Back at Home on Rogers TV and hear the stories from the towns and villages in New Brunswick that make this place our home.
Heartland to me is about the history.
Hosted and produced by people who live here, Back at Home brings you programming about New Brunswick directly from New Brunswickers.
The Harvey Fire Department is a big part of the community.
Back at Home is TV access to your community, exclusively on Rogers TV.
Inspiration might begin with a dish that is flavorful, sharing ideas that are resourceful, and full of tips that are insightful and beautiful. Wouldn't that be wonderful? Introducing Makeful, a new TV channel celebrating the creativity within us all. From food and design to DIY and style, get inspired by our exclusive series and passionate community of makers. Makeful, inspiration in the making, now in free preview.
Mosquitoes aren't that bad today. They were a lot worse the last couple of days. What are we gonna find on these? Any predictions? Any more slugs? What kind of snails? Robert? 21 meters? Okay. Should be able to start to see the flag too on the tree.
More to the left?
Yep.
Okay.
One board. Okay, there's one. And the third one? Okay. So which one are you guys going to pick? Bioblitzes are usually pretty short. This one here in New Brunswick is 10 days. And I've been really excited to be involved in this. Now, the slow revealing. Let's see who's under here. It's going to take a while, but it's not too many leaves. Some of the other ones have a lot of leaves on them. So you got to look each and every leaf just to make sure there's nothing there. And after a while, you get what's called a search image. So your brain automatically cues in on those things that could be snails. And we pick it up and, oh, we got another, oh, we got 3 slugs, including one orange one here, guys. Oh, he's even out crawling, you can see the tentacles. Nice. Small little tiny snail here. And Greta, you want to put it in your vial, or should I start another vial here? Okay. Little tiny fellow. We all realize that we live in this big wide world, and the economy is important, and forestry in some of the jurisdictions across this vast country are important. But at the same time, I'm hoping that the people that draw the lines on the maps, that cut down these trees, maybe they have a second thought. Are there any special species that are just found in this one little woodland? Maybe we should avoid cutting that woodland if it's habitat to one of these rare and endangered species. Yeah. Pulled in, yep, as soon as you touched them with the forceps.
I think that every single species on the planet is very important. So if it's a very tiny gastropod or an ant or something else, every single species is important, and it's important to protect every single species. So that's why we come out here and want to see what we have here, to already know what biodiversity exists, and then we can have any strategy to protect biodiversity.
Another specimen for the jar. Oh, two of them. Okay, good. Good, yeah.
I'm looking specifically on the effects of climate change. So I'm studying cold hardiness or heat tolerance. But in general, we can see that climate change affects snails because they're slow moving, they're always in the same habitat and need moisture. And so when climate change happens, then sometimes they have physiological limits. So then they're affected and can disappear from some habitats.
The most important thing you can learn from the humble mollusk, the land snail, is the quality of the habitat it's living in. I come from BC, and there's a lot of clear-cut logging and that sort of human-affected changes in the forest. And you can tell from years later what's happened to that forest by what's there as far as snails now.
Oh, a nice slime trail from these slugs here. No, looks clean. Bioblitzes are all about biodiversity. So how everything out there, even the smallest lichen, the smallest snail, the smallest slug, is important to the web of life. Usually you pull a string and you don't really know how that web of life will change. That's a nice little slug. It's one of the invasives again. That's a big problem in some of these forests when you get the invasive species coming in, and they're the bad guys. And one of the most interesting things that I've found here, the first day we were out, we were taking a look at some of the shells that you find along the lakes. And I saw this huge shell, and I said, uh-oh, I hope I'm not right. But later on, when I showed these shells to Don McAlpine, he said, yep, you're right. This was an invasive species. Previous to us finding it on Monday, it had only been found in a couple lakes down by Sackville, so lower Sackville, way down south in New Brunswick. And now it seems that this same invasive species is found in the St. John River system, which could be bad news for the native species that are found in the lakes. What this means is that we no longer have a very pristine area. When you have invasive species, they can compete with the native ones. And depending on the species that you're talking about, it could completely change the ecosystem.
This is the first occurrence of this gastropod in the St. John River system. I'd like to say it's going to be the last, but I'm afraid not. It seems to be reasonably well established here. So this is something that we'll need to monitor over the next few years to see whether it spreads or not.
It's pretty awesome being here in my own backyard and doing some of the research on animals that I've seen growing up and at home. Also, the kind of work I've done has been quite different, so I've learned a lot of really cool techniques in terms of sampling animals and studying different aspects of the organism's natural history and their behavior in life. Especially in terms of the Kichard fungus. I've never tested for Kichard before, and I've never actually collected samples testing for Kichard fungus. And it's a huge problem, especially in And a lot of herpetologists, a lot of people studying amphibians are now starting to turn their focus towards this disease and its impact on amphibians. So I've learned a lot of useful methods and I've gotten a lot of useful experience here, especially in my field.
This log looks good. Do you want to help me look over under this?
Yeah, sure, man.
Salamanders. They're the most abundant vertebrates in New Brunswick and eastern North America. So they play an important role in leaf litter ecology as far as salamanders go. Also, frogs play an important role of, controlling pests and the like. I'm having a lot of fun out here catching frogs and salamanders, just like I did when I was a 7-year-old boy. So, yeah, I've got no complaints. So when I'm out in the field, it's like I'm a child again, but it gets a little bit more sophisticated when we get back to the lab. We're screening all these amphibians for Kichard Fungus, which is a disease that's been wiping out entire species and driving major population declines around the globe. Our understanding of the extent of this, or really even the presence of that disease here in New Brunswick, is unknown. So I'll be going back to the institution in Oklahoma where I'm working and be analyzing this disease data that I've collected here in the PNA. This is a maritime garter snake, so it's a subspecies of the widespread garter snake complex.
Amphibians are bioindicators generally. They're very useful for us to study aquatic ecosystems and to know the health of an ecosystem, because amphibians are often impacted firsthand. So if you have amphibians in that ecosystem and you have the kind of baseline data that we're trying to obtain during this Bioblitz, then you can learn in the future if something is happening in that ecosystem, perhaps pollution or an introduced toxin or an introduced species will have a massive impact potentially right off the bat on the amphibians. So having them here in these protected natural areas and us knowing how many are here, what to expect in the future, will definitely benefit us.
The idea of losing any species is, for me personally, disturbing, not just because of the important role that amphibians play, but just because of their, my inherent interest and desire to maintain biodiversity. So it was discovered in the protected natural area, Grand Lake Meadows, and it will become a voucher specimen at the New Brunswick. So this Kichard fungus is not a natural thing in the environment. It's the result of humans spreading frogs around the world and thereby creating a hybridized strain of this fungus, which is really what's driving the decline. So it's a human-caused phenomenon. So we should take responsibility for it and do what we can to understand the disease and how to prevent, further extinctions. That was a good site.
Yeah, it wasn't bad. We didn't find our two-line, but
this wasn't the right habitat for those guys.
It's kind of a shame.
Yeah.
1, 2. Good rhythm back and forth. Hey Taylor, I think you might need this.
Thanks.
Rebound, scores! Taylor Hall!
Did I ever tell you guys the story of how
That guy almost ripped my shirt off.
I come here to drown my sorrows. You're fresh. I'm gonna cut some pancetta. Am I right, bro?
I can't decide how many powers you'd need to have to be more important than folk music.
I'm just going to saw this log while you continue to talk. I've been through a lot of accidents in my life, almost more than I can tell. And scene.
Cheese Monkey Box Pirate Improv, Wednesdays at 10 p.m.
Are you ready for this? Press the power-up chevrons. We got an alpaca. Hey, come on, that's mine. Why are you so angry all the time? Got you this pizza shaped like a...
What's so bad about my game?
Nothing. It's amazing.
Pause.
The village of Gagetown has worked extremely well for us. The people of the village have been very welcoming, very, very helpful. happy to have us here, and we're happy to be here. In most cases, the sites that we're working in are quite remote. The communities are dispersed. In this particular case, the village on the border of the PNA, it provides us with much easier access to some of the support services that we need to run a program like this. So it's worked very well for us.
Welcome to the New Brunswick Museum Annual Bioblitz.
This one's called the freckle pelt. You see it's got these little dark freckly spots on it.
One thing that I found that was really neat this time around was I found this little tiny mushroom, and they call it a mycena, and it's red.
The open house is an opportunity for the community, primarily adjacent to the protected natural area, to come into our field lab and to really share in some of the discoveries that have been made, to talk to the researchers, to hopefully discover some new things about these important conservation areas that are on their doorstep. Most of our focus here is, throughout the 2 weeks, is really on collecting data, and we, as much as possible, try to leave our researchers to do that. But we also recognize there's an important stewardship aspect in the work we're doing, and we really want to communicate that this new information that we're collecting with people who live close to these protected natural areas.
It's an incredibly important day for sharing everything that we do as biologists with the public so that they understand the importance of what we're doing and get people excited, and hopefully younger people excited about a career in biology, but also everyone else just to maybe step back and appreciate what they have in their own backyards. It's very important.
Science, of course, is one way to look at the world, and that's the one that we're really focused on, but it's not the only way. And in the second year of our program, in 2010, we started an artist in residence program, partly to provide that other perspective on the natural world, but also to document the work that we're doing. So for the last few years, we've had a number of artists. We've had Lita Karstead, who's a well-known wildlife artist, plein air painter from Ontario, who's been involved with us. We've also tried to get artists from the local community. And so they're doing painting in the field, sketching, drawing of the various habitats, the researchers, and the organisms themselves.
As you can see, I like to capture the people and what they're doing, as well as actually their findings, just to be with them and to watch exactly what they're doing. And I like to, again, I like to paint what people are passionate about and them enjoying their passion. The open house has been a wonderful event. Nice to have the people that have been supporting us in the community all along come and see what we're doing, and the children, of course, but the adults, and just to learn what the environment is like around them, because until my first year last year, I had no idea of what I was walking amongst, you know, the land and sea and everything around us. It's just, it's been an amazing eye-opener for myself.
You didn't pick any of these up, I don't think, in the previous...
No, it's the first time I've seen them here.
Yeah. In addition to our experts, we also have a fair number of students involved. And the Bioblitz provides, I think, a really unique and important opportunity for many of these students to get hands-on field experience and to work with really world-class experts. So it's a terrific experience for them. We've been at this for 6 years now. We've had a few students that have come up through the ranks. Some of them have started off as undergraduates, and we have one who's now going on to graduate school to study ants, who actually got most of his field experience here on the Bioblitz program. We've had a couple of others who've started off working on other groups of organisms and have continued with that interest in terms of developing a career. So I think the Bioblitz has an important impact for many of these students.
And coming towards me a little more. It's been a great experience. I've gotten to do work in with a variety of taxa in a variety of environments. Last year I was working with the mammal crew. This year I'm out with the fish crew, and it's been a great learning experience for me. I will admit, I didn't even want to touch a fish when I started this, and I've come a long way. I can tell, you know, different fish species now. I know the different methods of catching them. I'll get my hands dirty. And I've been really enjoying the experience. I love being out on the water. It's a great learning experience. It's a great job.
Well, this is my first Bioblitz, and I'm incredibly amazed by this experience. The experience I've gained and what others are gaining from being around other biologists in different fields, learning what other people are doing. We could be out in the forest looking for frogs and stuff, and for example, we found some crayfish, and I was very interested in them, and found some with eggs on its belly. I don't know much about crayfish, so we brought the specimen back, and we had somebody tell us all about them, about this neat parasite that they have that's only on crayfish. So the actual collaboration between biologists, and also you have a lot of senior biologists, a lot of biologists that are very affluent in their field, and then you have a lot of younger biologists such as myself, or students such as myself, and we're learning an immense amount from these veterans.
I have an opportunity to work with and learn from some of the best specialists in their own respective fields. I have an opportunity to further my education through professional development as well, and I have an opportunity to gain hands-on empirical experience by doing field work and by doing lab work here at the Bioblitz.
Being able to spend 2 weeks in the field with such a variety of experts has been just incredible for me. This is my 6th year doing this, and I've just learned so much and really learned how to appreciate all the rigors of field work and how to study all these organisms.
Especially I want to thank our participants. I want to thank all of you who've turned out this Bioblitz in 2014. It's always a challenge to raise the funds for this program. We've had the support of the New Brunswick Wildlife Trust Fund right since the beginning, since the first Bioblitz was run in 2009. We're very grateful for that, but it is every year a challenge to find the funding that we need to do this.
Hopefully today's program gave you some insight into the projects funded by the New Brunswick Wildlife Trust Fund. I'm Tom Benjamin. Thank you for watching.